Okay, so welcome to this first video in the playlist on noradrenergic neurotransmission. In this video, what we're going to talk about is the adrenoreceptors, okay, which are they're all G-protein coupled receptors. Okay, so I'm also going to put this set of videos in the playlist on G-protein coupled receptors. Okay, right. So we're going to start off by talking about the ligands for the adrenoreceptors, which are the catecholamines, adrenaline and noradrenaline. Okay, then what we'll do is we'll look at the adrenoreceptors themselves, and we will see how they're G-protein coupled receptors within the rhodopsin family of G-protein coupled receptors. We'll then have a discussion of heterotrimeric G proteins which they're going to interact with, and then we'll go through all of the different types of adrenoreceptors. Okay, now, firstly, just a bit of terminology. Some people call these adrenoreceptors, some people will call them adrenergic receptors, some people will just call them adrenoceptors, like I've done here. Okay, so they're all the same thing. I think this one is the most common nomenclature for the receptors to noradrenaline and adrenaline. Okay, so let's talk then now about the structures of noradrenaline and adrenaline. So both noradrenaline and adrenaline are called catecholamines. Okay, and I want to explain what we mean when we say catecholamines. So basically, catecholamines are all based on a structure called a catechol ring. Okay, so we'll start off with the structure of a catechol ring. So a catechol ring is basically a benzene ring. So we'll start off with a 6-membered carbon ring here, like so, and then we've got alternating double and single bonds, and then you have 2 alcohol groups coming off this benzene ring, and they are adjacent to one another. So here's the first alcohol group, here then is the second alcohol group here. Okay, so basically it's a phenol ring with an extra alcohol group on. So when you have a benzene ring with 2 adjacent alcohol groups coming off, that's called a catechol ring. And I want to stress that they must be adjacent. You can't have this alcohol group over here, for instance. That's called something different. It has to be next to one another. Okay, so basically adrenaline and noradrenaline both are catecholamines because they contain this catechol ring, and they also contain a side chain which has an amino group. Okay, so let's now see the structure of noradrenaline and adrenaline. So what I'm going to do is I'm going to flip this catechol ring around now. So we'll have it this side this time. Okay, so here's our benzene ring, alternating double and single bonds. Then we've got alcohol group off here and an alcohol group off here. And now we're going to have an amine side chain, basically. So what you'll have is then a carbon with a hydrogen coming off it and an alcohol group coming off it as well. Then you'll have your next carbon, which has 2 hydrogens coming off, and then it ends with an amino group here. Okay, so this then is the structure of noradrenaline here. So this is noradrenaline. I should just say that noradrenaline is the old name for this. There is a new name that is gradually gaining favour, okay, and that's to call it norepinephrine. Okay, so this new name is gradually taking over, but you will still hear noradrenaline used often as well. So norepinephrine. Right, so that's noradrenaline or norepinephrine. What we now want to see is adrenaline or epinephrine. And basically the difference between noradrenaline and adrenaline, or norepinephrine and epinephrine, is that adrenaline/epinephrine is going to have a methyl group coming off this amino group. Okay, so let's draw this. So let's start off with our catechol ring again. So here's this 6-membered ring with alternating double and single bonds, like so, and then we've got these 2 adjacent alcohol groups here, 1, 2, and then we've got our amine side chain here. So we've got this 2-carbon side chain here. There's the first carbon, here's the second carbon. Okay, and it's exactly the same, except now when we come to this amine group, we're going to have one hydrogen coming off it, and then we're going to have one methyl group coming off it here. Okay, and this is the structure of adrenaline, okay, which is also called epinephrine. So I'll put forward slash, and I'll put forward slash there as well. It can also be called epinephrine. Right, okay, so both of these molecules are ligands for all 5 types of adrenoreceptor we're going to see. However, there is more than 5 types, as we'll see. There's actually 6 alpha receptors and then 3 beta receptors, so there's overall actually 9 adrenoreceptors. Okay, so hopefully you understand why these are called catecholamines now. So they've got the catechol ring, both of them, and then they've got an amine group here, basically, and that's why they're catecholamines. Right, so what we're now going to move on to is talking about the adrenoreceptors then. Okay, now the adrenoreceptors are all G protein coupled receptors, so they're all GPCRs. Okay, and GPCR is just short, for G protein coupled receptor. Okay, now why are G proteins called G proteins? Well, G is short for guanine nucleotide binding regulatory protein, basically. So that's what G protein is short for. Okay, and we'll see how they are indeed guanine nucleotide binding regulatory proteins in a moment. Okay, so GPCR is just short for G protein coupled receptor. And strictly speaking, you should have dashes in between G and protein and coupled. Okay, right. So let's have a look at the structure of a G protein coupled receptor. So if this is the plasma membrane here, so I'll let these 2 lines represent the outer leaflet and the inner leaflet of the phospholipid bilayer, which means that this side is the extracellular fluid side, and this is the cytoplasmic side here. Okay, basically, G protein coupled receptors all have a characteristic structure, okay, and this structure contains 7 membrane spanning alpha helices, like so. Okay, so 1, 2, 3, 4, 5, now 6, and then 7. Okay, so the amino terminus of the G protein coupled receptor will be extracellularly, and the C terminus of the G protein coupled receptor will be intracellularly. Okay, so this is a structure that all G protein coupled receptors share. Okay, now G protein coupled receptors is the biggest family of proteins we know of. In humans alone, there are 800 different G protein coupled receptors that are known. Okay, so this is a massive, massive great family of proteins, and it's also extremely pharmacologically important because 30 to 40% of the drugs that are used clinically work by acting on G protein coupled receptors, and we're going to see many examples of drugs that are used clinically which work on the adrenoreceptors specifically. Okay. Now, G protein coupled receptors are split into 5 separate families, okay, and the main family of G protein coupled receptors is the first family that we'll discuss here, so family number 1, and this is the rhodopsin family of G protein coupled receptors, named because it contains rhodopsin, which is a photoreceptor used in the eye. Basically, it's a receptor that responds to light. Okay, so this is the rhodopsin family of GPCRs. Basically, this is easily the biggest family of the 5 families. Okay, it contains around 750 of the 800 members of the total G protein coupled receptor family. Okay, so it's easily the biggest. And basically, the characteristic feature that all the rhodopsin family G protein coupled receptors share is where they bind their ligand. Okay, so let me show a G protein coupled receptor here. Okay, so rhodopsin family G protein coupled receptors all have very small amino terminal domains here. Okay, and that's because they don't actually need the amino terminal domain for binding their ligand. They will bind their ligand to the transmembrane domain, basically. So if I get my blue pen here, this blue dot here is meant to represent the ligand. Basically, the ligand for rhodopsin family G protein coupled receptors binds to residues that are in the 7 membrane spanning alpha helices. Okay, so it binds to the transmembrane domain. Right, and that's the characteristic feature of these rhodopsin family G protein coupled receptors, and all of the adrenoceptors, which are the topic for this video, are within this family. So they're going to bind their ligand in the transmembrane domain. The noradrenaline or the adrenaline will bind to residues which are in these 7 membrane spanning alpha helices. Okay, and generally it's in the extracellular third of the membrane spanning alpha helices that the ligand binds, i.e. it doesn't come right down here and bind. It's generally in the extracellular third of the receptors. If I just sort of put a line across there, this is the extracellular third of the membrane spanning alpha helices, and generally the ligand will bind to residues within that portion, rather than the intracellular 2/3 of the membrane spanning alpha helices. Okay, right, so just for a complete picture of G protein coupled receptors, I'll continue on and talk about the other 5 families, although adrenoreceptors all fall into this first family. Okay, so we'll continue on. Family 2 then. The second family of G protein coupled receptors is what's known as the secretin family of G protein coupled receptors. Okay, now the secretin family of G protein coupled receptors contains many receptors which generally have as their ligand peptides. Okay, so the ligands for these receptors that are in this family are generally peptides, but that's not the characteristic feature that they all share. Basically, the characteristic feature that they all share is that they have a quite large amino terminal domain, which I'll show here. Then they have the 7 membrane spanning alpha helical domain. Okay, there we go, and I'll put the membrane in here. So here's the amino terminus over here, and here's the carboxylic acid terminus down here. And basically the characteristic feature that all of these secretin family G protein coupled receptors share is that they bind their ligand between the amino terminal domain here and the transmembrane domain, so it gets sort of wedged in this gap between the amino terminal domain above and the transmembrane domain below. Okay, and that's the characteristic feature of secretin family G protein coupled receptors. Now let me give you some examples of receptors that are in this family. Okay, so for instance, the receptors to calcitonin are within this family. The receptors to parathyroid hormone, both of these two are peptide hormones, and they're very important in the regulation of calcium level within the blood. Okay, receptors to glucagon, another peptide hormone, are examples of secretin family G protein coupled receptors. Okay, so these all abide by this general rule here, this general structure. Okay, right. Next family of G protein coupled receptors, family 3. Okay, so the next family is what's known as the glutamate family, and you might be able to guess which receptors this is going to contain. Okay, so basically the characteristic feature that all glutamate family G protein coupled receptors share is again a fancy amino terminal domain structure. Okay, so they have this sort of domain here. Okay, and then they have the 7 membrane spanning alpha helices, like so, and then here's their carboxylic acid terminus down here. Basically, this is the fancy domain that the glutamate receptor family G protein coupled receptors all have, that I'm now highlighting in turquoise. Now what's this called? Well, this is called a venous flytrap domain. Okay, and basically for glutamate family, I'm about to write family, venous flytrap domain. Okay, so for glutamate family G protein coupled receptors, the ligand binds in here, basically in this venous flytrap domain, and the idea is this, that when the ligand binds, it's like a fly going into a venous flytrap. Basically it starts off open, and then when the ligand comes in, it closes around it, basically. And that's the sort of domain that all of these glutamate family G protein coupled receptors have in their amino terminal domain. Okay, So, what examples are there of glutamate family G protein coupled receptors? Well, most glutamate receptors in the brain are not G protein coupled receptors. Glutamate is the main excitatory neurotransmitter in the brain. So when you have one neuron synapsing onto another neuron, and this neuron, well, the presynaptic neuron wants to trigger an action potential in the postsynaptic neuron, usually they release glutamate and that excites the next neuron along. Okay, so that's what's happening in the brain. Now, that excitation is not achieved through G protein coupled receptors generally. Instead, it's achieved by ligand-gated ion channels. So most of the glutamate receptors are ligand-gated ion channels that will open and allow sodium ions to enter the cell. So for instance, the AMPA receptors, the kainate receptors, the NMDA receptors, these are all examples of glutamate receptors that are ligand-gated ion channels. Okay, so they're not G protein coupled receptors. However, you do have some glutamate receptors which are G protein coupled receptors, and these are called the metabotropic glutamate receptors, and there's around 8 of them. Okay, so this little M here stands for metabotropic. Okay, and metabotropic receptors is another name for G protein coupled receptors. So this basically says the G protein coupled receptor glutamate receptors. Okay, that's what this stands for. And as I say, there are around 8 of these called mGluR1, mGluR2, mGluR3, all the way up to mGluR8. Right, so those are examples of the glutamate family G protein coupled receptors. There's another very famous example within this family, which is the GABA B receptors. Okay, so glutamate is the main excitatory neurotransmitter within the brain. GABA is the main inhibitory neurotransmitter in the brain. So if the presynaptic neuron wants to stop the postsynaptic neuron from firing an action potential, it will secrete GABA onto it. Now again, the receptors for GABA, the normal receptors for GABA, are not G protein coupled receptors. Okay, they are instead ligand-gated ion channels, this time of the cis-loop ligand-gated ion channel family. Okay, and those allow chloride anions to enter the cell and hence hyperpolarizes the cell membrane and makes it less likely to fire an action potential. Those are the GABA A receptors that are ligand-gated, cis-loop ligand-gated ion channels. Okay, there is, however, this second family of GABA receptors called the GABA B receptors, which are G protein coupled receptors, and they too are in this family of glutamate family G protein coupled receptors. Right, okay, so the fourth family of G protein coupled receptors then. So the fourth family of G protein coupled receptors is the adhesion family of G protein coupled receptors. Okay, and basically what's characteristic about these G protein coupled receptors is that they have a large extracellular domain, so a large amino terminal domain here, and they need this for binding to their ligand, which is not a conventional ligand. Instead, their ligand is a component of the extracellular matrix. Okay, so here's their amino terminal here, here's the carboxylic acid terminal, and basically their large amino terminal domain here will bind to components of the extracellular matrix. So in turquoise here, this represents a portion of the extracellular matrix. Okay, so these adhesion family G protein coupled receptors, they all bind to components of the extracellular matrix. Okay, and then we'll go on to the final family, which is the family with the stupidest name. Okay, so this is the frizzled / taste 2 family. And this is named after the two major members that are within this family, okay, which are the frizzled receptor involved in the Wnt beta-catenin pathway and the taste2 receptor involved in the gustatory system. Okay, so basically this family kind of contains everything that doesn't fit into one of the other families. Okay, so they bind their ligand extracellularly, so they have an amino terminal domain that then helps them bind their ligand, but they don't have any of the interesting features that the others have. Okay, so they can't be rhodopsin family because they bind it by the amino-terminal domain, not the transmembrane domain. They can't be secretin family because they don't have this property where the ligand binds in that slot between the amino-terminal domain and the transmembrane domain. They're not glutamate family because they don't have the venous flytrap domain. And they're not adhesion family because they don't bind components of the extracellular matrix. So their ligand just sort of binds to the amino-terminal domain and activates the receptor that way. So here's the ligand in blue. Here's their carboxylic acid terminus here. And as I say, the two major examples of this are the Frizzled receptor, the ligand for which is Wnt, and the taste-2 receptor, the ligand for which is bitter-tasting molecules. Okay, so that's the 5 families of G-protein coupled receptors. Okay, right. So, basically, all of the adrenoceptors are members of the rhodopsin family of G-protein coupled receptors. Now what I want to do is tell you about all the different adrenoceptors that we have, and then what I want to do is go over heterotrimeric G proteins, and then I want to go through every single one of the receptors, or at least every one of the categories of receptors, in turn, and look at what they actually do and the signaling pathways that they activate. Okay, right. Let's start by just naming the receptors. So, firstly, the first division of the adrenoceptors is that they can be divided into the alpha receptors and the beta receptors. Okay? After that, you can then divide them up into alpha 1 and alpha 2, and then beta 1, beta 2, and beta 3. Okay, now some people think there's a beta 4 as well. There is growing evidence that there's a beta 4 that's on the heart, but we won't go into that because so little's known about it anyway. Okay, right. So, that looks as though there's 5 separate receptors. There's the alpha 1 receptor, the alpha 2 receptor, the beta 1, the beta 2, and the beta 3 receptor. But since then, what's happened is that alpha 1 and alpha 2, we have realized that they are basically not just one receptor. There are multiple alpha 1 receptors which all do pretty much the same thing. So there is an alpha 1A receptor, there is now an alpha 1B receptor, then there's an alpha 1D receptor. Similarly, alpha 2, it's become understood that actually there's many different alpha 2 receptors. So there's an alpha 2A receptor, an alpha 2B receptor, and also an alpha 2C receptor. Okay, now these are not just different splice variants of the same gene. They are different genes that code for these different alpha receptors, basically. So there is the alpha 1A receptor, there's the alpha 1B receptor, there's the alpha 1D receptor, the alpha 2A receptor, the alpha 2B, the alpha 2C, and then beta 1, beta 2, beta 3. That takes us up now to 9 separate adrenoreceptors, and they are all rhodopsin family G-protein coupled receptors. Okay, so basically, what used to just be called the alpha 1 receptor, we've realized that there are actually 3 different types of this. Similarly, what used to just be called the alpha 2 receptor, we've realized that there are 3 different types of this as well. So that we now know that there are 9 different forms of adrenoreceptors. Okay, right. So, what we will do is we will treat all of these alpha 1 receptors as one, because they all do pretty much the same thing. We will treat all of the alpha 2 receptors as one, and we'll treat these ones. We'll kind of treat these together as well, because they all activate the same sort of pathway. Okay, right. So we'll call it there for this video, and we'll continue our discussion in the next video where we'll move on to discuss heterotrimeric G proteins.